Okay, so this is going to be the chapter 1 crash course. So, what are microbes? Where are microbes found? So, microbes are found basically everywhere. They're ubiquitous. They make up 50% of Earth's total biomass. So, what you need to know is that, you know, microbes are everywhere. Microbes are also synonymous with bacteria. Explain why there are different varieties and quantities of microbes in different locations and environments. So basically, microbes are everywhere because microbes can tolerate and prefer different temps, oxygen, pH, salt concentration, etc. So basically, a certain microbe, this one microbe, right, can prefer cold environments with low oxygen, low to no oxygen, lower pH, salt concentration, and etc. While this other microbe can thrive and grow in places of high temperatures, a lot of oxygen, high pH levels, and salt concentration. So basically, there's different microbes and bacteria that prefer this place to this place and can thrive in different places in different places. That's why there are so many different bacteria and why they are basically everywhere. So, what are the different subfields of microbiology? So, my mnemonic is Be My Valentine PP. So, that's kind of crazy, but it stands for bacteriology, mycology, parasitology, virology, protozoology, and phycology. The ones you really need to know is bacteriology, mycology, parasitology, and virology. Basically, study of bacteria, study of fungi, study of parasitic organisms, and study of viruses. Environmental roles. So basically, the microbes contain or make up of around 50% Earth's total biomass. Plants only 35, animals only 15. Basically, what you need to know is that microbes are everywhere, and they basically are very, very, very useful in sustaining life on Earth, right? And that gets to our next point, the environmental significance. So it aids in recycling critical elements is carbon, nitrogen, phosphorus, sulfur, and basically sustains life on Earth of different, microbiotas. So for example, there's a lot of microbes in your stomach that maintain the functions in your stomach. There's microbes in the soil that basically sustain life on Earth, etc., etc. Practical applications of microbiome. So, some practical uses are food production, right? So, Saccharomyces is a type of bacteria, which is baker's yeast that are used to make bread. Fermentation. Think about taking a cucumber, fermenting it into a pickle, and cheese making. Medical production. So, a lot of antibiotics are made from bacteria and are used to basically fight against other bacteria, right? And then why is it important to know about and understand what emerging infectious diseases are? So it's important to know about certain pathogens because it allows us to understand how to fight against them. So for example, this pathogen has different characteristics, like it's gram-positive, it's an acid-fast bacteria. We need to find a medication that basically fights against these characteristics, right? We need an antibiotic that is good against gram-positive bacteria that also can fight against acid-fast and yada yada yada. So basically, knowing what characteristics of a pathogen is allows us to accurately find medication to stop the spread of that disease. Getting into the scientists, you need to know Leeuwenhoek. Who is Leeuwenhoek? So basically, he is the first to describe bacteria. He also created the first simple microscope and is the father of microbiology. And I'll go more into him later. Another important scientist you need to know, Robert Koch. Koch made the Koch's postulates. He created streak plate method and was the first to isolate pure cultures. He also created germ theory, which linked single isolated microbes to a human disease. And I'll get into that later. So Louis Pasteur, another very important guy you need to know. Louis Pasteur, he basically disproved spontaneous generation by Swan neck flask experiment, and he also created pasteurization. Those are the big things that you need to know about him. He also proved that different strains have different properties, and also proved that fermentation is caused by microorganisms. Carolus Linnaeus, he's the father of taxonomy, created the first tree, animal, plant, and mineral. Whitaker, He added fungi to the tree, so now the tree is animal, plants, participant, prokaryote. Carl Woos, you need to know him. You need to know him. You need to know that he created the current tree of life, which is the 3 domains of bacteria, archaea, and eukaryotes, based off of rRNA sequences from DNA gene sequencing. Robert Hooke introduced cell theory and first to see cells. I didn't include this, but he also was the first to make compound microscopes. You need to know that. Semmelweis, hand washing. I'm not sure if Semmelweis or Listerine will show up on the test, but Listerine made carbolic acid and used it as antiseptic. And he's also that's what Listerine mouthwash is made after. More on Pasteur. He made the swan neck flask experiment. So basically, he heated up 2 flasks, and then there is like a swan neck curve, right? And then basically, bacteria was prevented bacteria from entering the broth and basically made it sterile, right? So basically, his theory was that if I were to leave this broth out, right, the sterile broth out, will it be contaminated after a period of time? Because that's what spontaneous generation is basically saying, is that, spontaneous generation is where, germs come out of nowhere. It just happens. You're just unlucky. You just got, sick. But basically, this curve prevented bacteria from entering the broth, right? And after he removed the curve, that is when air got into the broth and contaminated it, making it unsterile. So basically, after it sat for a while, it stayed sterile. But once he removed the curve and allowed air to get in, that is when the broth became unsterile and contaminated, disproving spontaneous generation. Koch's postulates, there's 4. So basically, it starts with an animal with a disease, and basically a healthy animal without the disease. So what happens is that we isolate the disease from the sick cow, and then we get that isolated disease, put it in a healthy cow, and see if that cow gets sick. So then the cow gets sick, the diseases were produced in the healthy cow, and then basically we have a sick cow, a newly sick cow, which means that by theory we should be able to get that disease again and recreate that process. So we get the disease from a newly diseased cow, get it, put it in another healthy cow, and then the process goes over and over and over again. And understand the connection between microbes and infectious diseases and how the discovery led to this. So basically, from that Koch's postulates, right, we are able to isolate these pure cultures through Koch's postulates and establish germ theory. And germ theory is essentially that germs cause certain diseases and is spread through a vector, not through spontaneous generation. So from Koch's postulates, we know that this certain germ caused this certain disease, and it's not through spontaneous generation, but through a vector. And one of these vectors could be something that could hold or carry disease, like dirty water, contaminated food, rats, dogs, and etc. And those vectors are what spreads the disease. And 3 domains of life, which is created by Woese again, bacteria, archaea, eukarya, built off of DNA sequences off of RNA sequencing. What is taxonomy? Taxonomy was created by Linnaeus and is basically a system of naming and categorizing organisms based off of relation. And why are viruses not on the tree of life? So viruses are not considered on the tree of life because it is not living, because it's acellular and non-reproducing. That's what you need to know. So explain how the growing body of knowledge about living things contributed to the development of increasingly complex trees of life. So basically, over time, the tree of life started with Linnaeus to Woese, and basically evolved and simplified to accurately categorize living things based off of shared characteristics. 3-domain system. Like I said, bacteria, archaea, and eukarya. That's it. What are the taxonomic hierarchies and who established these? So these were established by Carolus Linnaeus again. And so it is from the broadest domain and it goes all the way down to specific. So domain, kingdom, phylum, class, order, family, genus, species. And you can have your own mnemonic for that. Understand how nomenclature of living things work. Rules of nomenclature. So basically it's genus and species. Right? It's made by Carl Linnaeus. And basically it's genus then species. So for example, Clostridium difficile, C. diff. So genus is the more broad term, and then species is the more specific term. And species versus strain. So basically, we can have Clostridium difficile, so C. diff, and species is basically that, right? So we have a species of C. diff, and then a strain. So C. diff 12. So that's a genetic variant of that species, which either means it's more pathogenic, so more deadly, or could be less deadly, yada, yada, yada. Bergey's manual is basically the information for classification, identification of bacteria based off characteristics. So it's like basically classifying bacteria based off whether it's like, what's it called? Aerotolerant, aerobic, stuff you don't need to know yet, but basically, just based off of characteristics. Bacteria. So it's a single-celled organism that lacks a nucleus, is prokaryotic. You need to know that. Viruses, acellular. Infectious agents that require a host to be replicated and is made up of genetic material, so DNA or RNA, and basically causes diseases, right? Okie dokie, eukarya, protists, algae, and fungi. You don't really need to know about archaea that much. And some prokaryotic and eukaryotic acellular microbes with examples. So Naegleria is eukaryotic brain-eating amoeba. Giardia, eukaryote that causes diarrhea. Candida, eukaryotic fungi that causes yeast infections. Saccharomyces, eukaryotic fungi used as baker's yeast. E. coli, prokaryotic bacteria that makes your tum-tum hurt. And then relative size, you need to know this picture really well, or at least know that from smallest to largest. Lipid is going to be the smallest, right? Then virus, then bacteria, because viruses will always be smaller than bacteria, to red blood cell, and human egg or frog egg. That's all you need to know. Prokaryotes. You need to know the difference between prokaryote and eukaryote. Okay, so prokaryotes, unicellular. Need to know that. Non-membrane bound nucleus with single circular chromosome located in nucleoid and 70S ribosomes. You need to know that on the test. Eukaryote is the opposite. So it could either be multi or uni, but it is membrane bound with 80S ribosomes. And then that's it. Good luck.